Thanks for the organizers for allowing me to present my work on derivation of macaque lung progenitors from pluripotent stem cells as therapeutic agents after lung injury. My name is Anna Engler. I'm a postdoc at the Center for Regenerative Medicine, and I'm a member of the Chapman Hub. Previous work from the SNUC lab, but also the Cotton lab, has shown that induced pluripotent stem cells from humans can give rise to lung progenitors if they're manipulated correctly. And these lung progenitors can then be pushed towards either alveolar progenitors or airway progenitors given high or low wind, and they can be used for disease modeling, but also genetic permutations. So one idea is also that these cells could be taken from a patient, let's say with cystic fibrosis. The donor cells are then genetically gene edited to become a wild type or a healthy cell. And one could use these cells as transplantation agents, so a cell-based therapy after lung injury. However, there is a very big ethical concern right now that human-to-human experiments are not possible. And so we need a large animal model as an alternative to show these proof of concept experiments. And we have gone to a very close relative of humans, which is the rhesus macaque monkey. And we are now trying to recapitulate these results in macaque iPS cells. So to give you a bit of an outline of my talk, I'll be first talking about how we've established a protocol to get macaque basal cells, and we've characterized these cells in great detail to have a hallmark for then our directed differentiations towards lung progenitors. And then as a third part of my talk, I would like to talk about the various injury models that we've looked at that we hopefully can then use to take these cells, these directed differentiated cells, to transplant them into these injured lungs and hopefully improve injury repair. So firstly, we needed a protocol where we can get a control tissue, which are the basal cells. And for that, we used upper airways from rhesus macaque lungs. These were then digested overnight in a very gentle digest buffer, scraped off, and within 24 hours, we see these basal cells that are attaching. And we can passage these cells for 10 passages, and they maintain their keratin 5 and P63 identity. So these are basal cells for about 10 passages. And this protocol has been published last year. So if you have any questions, this is where you can read about it. We then used these cells, and this was done by Shingo Suzuki in the Davies lab, to target them with an NKX 2.1 GFP construct. NKX 2.1 GFP is a marker that is expressed in the lung, the thyroid, and the brain, and it's generally used to as a reporter for lung specification. And we were able to target these basal cells with the NKX 2.1 eGFP construct, and we can sort out these GFP positive cells and maintain them as an almost pure culture for a long time. We then used these GFP targeted cells and their wild type counterparts to generate macaque iPS cells. And we use Sendai virus containing the Yamanaka factor, and we are maintaining these basal cells on feeders. And all the lines that we're using have been karyotyped, and they're karyotypically normal. So as a first thing, we wanted to see whether we can maintain these cells, but also potentially direct differentiation. And we got inspiration from nature for this. So these iPS cells are plated on feeders and maintained in high levels of FGF. And we're then using the stem cell technologies endoderm induction kit to get endoderm from these macaque iPS cells. We then use dual SMAD inhibition conditions to go towards a foregut fate. And then from this foregut point, we are using Wnt and BMP signaling to go towards a lung specification. And once we've achieved this lung specification, we're hoping that using high FGF or high Wnt conditions, we can go either to a proximal or a distal fate. Firstly, our macaque iPS cells express stemness markers such as TRA160, 81, and SSEA4 in our maintenance conditions. However, they do not express markers of differentiation such as SSEA1. When we then use these cells in an endoderm induction using the STEM-diff kit, we get between 50 to 70% of endoderm induction, seen by CXCR4 and c-KIT on the protein level, but actually also SOX17 and FOXA1 on the RNA level. We then tried to take these cells and put them through the human protocols that have been published for lung induction. And what we see is, using the human protocol, we actually only got very little NKX 2.1 GFP expression. So what we're currently in the process of doing is we're trying to now optimize the protocols to have a Macaque protocol to get lung differentiation. And we're able to get a bit higher NKX 2.1 numbers with the NKX 2.1 GFP. And these cells also on the RNA level express NKX 2.1 for the lung, but are devoid of markers such as PAX6 or PAX8, which would mark neuronal or a thyroid fate. So we are still optimizing this protocol, but once we have these cells, we want to have everything else in place. So I want to briefly touch upon the injury models that we've looked at. We've used various injury models in the rhesus macaque. However, there was one hallmark that we were specifically looking at, which is this ectopic keratin 5 pods that have been previously published from the Chapman lab. And there was actually only one injury model that gave us these pods, which is bleomycin. So the animals obtain bleomycin. And then at 3 weeks, we see these keratin 5 positive pods that we're hoping we can then take our lung progenitors that we're deriving from the iPS cells. And then these progenitors hopefully will integrate into these keratin 5 positive pods and hopefully speed up regeneration and repair. With this, I'm at the end of my talk, and I would like to summarize. We're able to maintain, isolate, characterize, and differentiate macaque basal cells. We were then able to target these cells with an NKX 2.1 GFP construct and maintain them as iPS cells on feeders. These cells are karyotypically normal. We can induce endoderm and have started to optimize the lung induction given with an NKX 2.1 GFP reporter line. And we're then hoping that we will be able to take these cells to improve the injury repair in rhesus macaque monkeys after bleomycin injury. With this, I thank you for your attention, and I'm happy to take any questions in the question session after.